Okay, ladies. Today we're going to talk about how. So we'll keep it short today because it's a short period. We'll talk about how neurons get triggered. So now we talked about how neurons work, and we talked about basically the movement of sodium and potassium in and out of neurons. And then last time we talked about communication between neurons, or neurons and muscle, for example, involving neurotransmitters, so chemical synapses. Today what we're going to do is we're going to talk about like triggers. So we'll look at a couple of examples of how different neurons get triggered, okay? So let's start off with a concept in when it comes to neurons, right? I want to show you this this data, okay? Now, take a look at basically this information. It says that if you take stimuli, right, and the stimuli has a voltage, so this is in millivolts, okay? You'll notice that at 1 millivolt, so that's the stimulus, okay? So the stimulus could be anything. So what do you think the stimulus would be for your cells that detect sound? It would be sound waves, right? It will be some disturbance in the air that those cells can detect, and we'll talk about that later on. What do you think the stimulus would be for touch? It would be what? Pressure. There would be pressure on your skin, right? And we saw the stimulus for smell. It's actually a molecule that interacts with a receptor. So here, the stimulus to cause contraction. So these are muscle fibers. So the neuron would secrete a signal. The signal would change the voltage, for instance. Now at 1 millivolts, what happens? Nothing. What about at 2 millivolts? Do you get contraction? Yeah, you do. 3, 10. Now, here's the thing. Does it change? So what you're seeing here is something called the all or none response. Okay? The all or none response says that a neuron is either going to fire or, guess what, Jerusha? It's not. Here's the other thing. If it fires, the action potential will look now we're going to fill in the blank. So, take a look at this. You see these action potentials? Okay. You see the red one? And you see the blue one? And you see the green one, okay? So let's say the red one is the action potential for the stimulus at 2 millivolts. So we know what an action potential is, the movement of sodium in and out, right? And that changes the voltage because those are positive ions moving in and out of a cell. Now, if we were to measure the height of the action potential for 2 millivolts, this is the height, okay? So it's from here to here. That's the height. What if we were to measure the height of the action potential for 10 millivolts? Okay, for 10. Would it be the blue line, which overlaps the red, or would it be the green line? What do you think it would be? Which one? What do you think? Do you even know what I'm asking you? Think of it like a tidal wave, okay? Think of this as a tidal wave. A wave is a disturbance, right? So imagine that we have a tidal wave. So here's my tidal wave. The height of the tidal wave would represent the energy of the wave. So if you were to think of these as little tidal waves, would a 2 millivolt stimulus produce a tidal wave that's bigger? The tidal wave is just the action potential, okay? Or would it produce a tidal wave that's exactly the same? What do you think the answer is? It's actually the same, and that's what you're seeing here. Take a look at what you're seeing. It's the same. So two things. Here's the two things. One is either it fires or it doesn't. That's the all or none response. Secondly, is if it fires, if the neuron fires, each action potential looks exactly the same. So then here's the question then. If each action potential looks exactly the same, how do you distinguish intensity of pain? So let me give you an example, right? If I prick you with a pin, okay? If I stab you with a little pin, would you feel it? Okay. So imagine you were measuring the little action potentials coming out of the neuron. It would look like this. Okay? It would look like this. Now, if I stabbed you with a knife, not that I would do that, because I'm on record here, okay? And it's totally illegal and, unethical for me to injure my students. But imagine I accidentally stab you with a knife. Would it feel the same? It wouldn't, right? But what if we were to measure the little action potentials from the neurons that detected the damage? Well, it wouldn't look like this. It would look like this. So whether the neuron was like detected the pinprick or whether the neuron detected the knife wound, the action potentials would be the same. But yet you can tell the difference between the two, right? So how do you do that? If the action potentials are the same, how does your brain know what's really painful and what's not so painful? What do you think the answer is to that? What do you think? So even though each action potential is the same, the number of action potentials isn't. So in other words, even though the strength of the stimuli does not affect the height of the tidal wave, it would affect the number of tidal waves. So a very intense pain would generate more action potentials. And your brain would be registering more action potentials and therefore would say, okay, this means more pain. Lower action potentials would mean less pain. Okay? I have a question for you. I want you to think about this, okay? Now, this actually has to do with not really today's topic, but actually has to do with something we talked about before. I want you to think about this, right? Right now, how many of you are thinking, and this, by the way, as soon as the words come out of my mouth, it will change everything. But right now, how many of you are thinking about how really cool it feels to be, or not how cool, that's the wrong word. How it feels, how the clothes on your body feel. That's what I want to say. Yeah. How many of you are thinking about how the clothes on your body feel? How many of you were thinking, before I asked the question, how the clothes on your body feel? You weren't thinking about it? Why not? Your body's covered in clothing. Can you detect the clothing that you're feeling? Can you feel it now? You're thinking about it now? How come you're feeling it now? Why? But it was there all the time. But why not? So what happened? What happened? Why weren't you thinking about it? Okay, how come right now, can you feel it? If I say to you, can you feel your clothes? How can you feel it? Right? And obviously what's happening is it's generating enough of a stimuli to do what? To trigger the neurons, the touch receptors that allow you to detect things on your skin, right? You ever have something that's crawling on your skin that you didn't even notice it was there? Why didn't you notice it? So it's putting pressure, right? It's putting pressure on your skin, but yeah, very good, Emily. There's the strength of the stimuli, which is the amount of pressure in this case that is pushing down on the skin, wasn't enough to trigger the neuron. Now you may have seen it. That's different, right? Those are different neurons seeing it. But you're close. I mean, you feel them, so you know you're getting the neurons are being triggered, right? Because you can be like, oh, I can feel my clothes. But why weren't you really thinking about it? What's happening there? It's actually very similar to what happens when you walk into a room. How many of you have ever walked into a room and you walked and you said, oh my God, it smells so bad in here. But yet the people that are in the room are like, what are you talking about? Now, one of two things. Either the people in the room don't have a good sense of smell, which is possible, right? But probably a better explanation is something else. Very similar to what we were just talking about. Yeah, so we call that sensory adaptation. So what do you think is actually happening from a chemistry point of view? So you feel your clothes, but at some point you get this feedback effect where chemicals will be released that will do what? What do you think these chemicals will do to those neurons? Do you think they'll make those neurons more active or less active? How? How will they do that? Will they hyperpolarize them or hypopolarize them? Will they push the voltage further away from threshold or closer to threshold? Remember we talked about two types of neurotransmitters, those that promote action potentials and those that inhibit. So like GABA inhibits because it makes the neuron more negative. What would be happening there? What do you think would be happening there? Let's go back to our picture of the action potential. If we can make these two things closer together, the neuron will fire more frequently. So what do you think? Oh, by the way, remember in the slide about the nervous system where it says that the threshold for something is lowered and you become more aware of your environment? So here's the thing. You see, this is the resting state of the neuron, and this is the threshold. Whether this goes up or that comes down, the closer these two lines get to each other, the easier it is for the neuron to fire. So what do you think is happening when you have sensory adaptation? When you stop thinking about the clothes that you wear, when you're sitting in a room that's smelly but you don't notice it anymore, what do you think is happening to probably the resting state? Which way is it moving? Up or down? Down. Moving down, okay? Moving down. I should have mentioned that last time when we were talking about neurotransmitters. By the way, when you have a fever, so you know how when you have a fever, and the fever goes really high, that can induce seizures? Now, when you have like seizures, basically the neurons are firing out of control. So in that case, what's actually happening is the increase in temperature is affecting the proteins in the neurons, which actually changes the threshold. Which way do you think the threshold moves? Which way do you think the threshold moves? Up would be further away than from resting state. Yeah, moves it down. So the threshold in the neurons when you have a high fever actually gets pushed further down, which means it's closer to resting state. So they're easier to fire, and that can cause seizures. Okay. All right. So let's talk about now the trigger mechanisms of neurons. Okay. So let's talk about triggers. So we're going to look at some examples of how certain receptors can be stimulated. So here's one. This is taste. Okay? You don't have to memorize all of the interactions. I just want you to understand like the big picture. Okay? So here's the big picture. One, all neurons behave the same way. So whether it's a neuron from a fruit fly or a camel or a giraffe or Jerusha, it's all sodium comes in, potassium goes out, sodium potassium pump restores it. Okay? The communication between the neurons is going to be the same mechanism. Neurons communicate with other neurons through neurotransmitters. Okay? That's the thing. But what's different is the way they get triggered. Okay? What's different is the way they get triggered. For example, this is the trigger mechanism for a neuron that detects what? Taste. Specifically what taste? Sugar. How does it work? What needs to happen here? Sugar has to interact with a receptor. What's the end result? So the channel for potassium will what? You can see closes. Remember what a protein kinase is? It's an enzyme that adds what? Phosphate. When we add phosphate to proteins, it changes their shape. Okay? That's an important thing in cell biology. In this case, the addition of phosphate to a protein, the protein is a potassium channel. That's what it is. When we add a phosphate to that potassium channel, the shape of the potassium channel changes. It changes in such a way that it closes up. Okay, it closes. Now, here's the thing. Why would that trigger this neuron? Now, remember, there are two types of gates in neurons. There are the voltage gates. Actually, so if we remember the picture of the neuron, we'll go back. Where is it here? There are gates that are controlled by the voltage. So for potassium, the voltage gates, the voltage controlled gates for potassium will open when the voltage is positive, okay? And for sodium, the sodium voltage gate opens when it's about negative 55. But there are other gates, okay? Those aren't the only gates in neurons. So there are gates that are not controlled by the voltage. So this is an example of one. It's not controlled by the voltage. But why would this trigger an action potential? Well, I want you to think about this, okay? You have a neuron that the dotted line is the threshold. The straight line is the resting state, okay? Now, some potassium leaks out through this channel. So there are channels in the neuron that are open, that are not controlled by the voltage, where some of the potassium will leak out. But what would happen if we closed that channel? So no potassium leaks out. That means, what are you doing? You are retaining more what? Yeah, and what's potassium? What charges it? Positive. So what happens then over time is these positive charges start to accumulate in the neuron. That changes the voltage and drives it up. And then what happens when we get to that magical threshold? Boom. Neuron gets turned on, fires. The information gets sent to your brain, and you're like, wow, this is sugar. Which is, well, no, not bad, depending on the type of sugar, right? Depending on the type of sugar. If you're watching the video, they say fructose is bad, but, okay? So this is how this receptor works. But let's take a look at another receptor. Let's take a look at the odor molecule and how it's detected. It looks similar, right? It's you can see here, need a receptor. Here's the odor molecule, interacts with the receptor. But the difference is we're not affecting the potassium channel. We're affecting the sodium channel. Now, again, this is not a voltage-controlled sodium channel. This is a different type of sodium channel. What happens in this case? The sodium channel what? Opens. But look, remember, in a neuron, most of the outside, sodium is on mostly on the what? On the outside. So in this case, it's actually the same result because instead of keeping potassium, you're bringing in what? But it's the same thing, right? It's still an accumulation of what? Positive charge, which then raises the voltage to threshold, and then you're on fires and you detect that molecule. Okay? You detect that smell. I'll come back to like drugs and chemicals. Your whole lab was on that. We'll come back to that later on, okay? I'm going to skip over that. I'm going to skip over this because today I want to focus on just some examples of sensory receptors. So let's talk about, for example, vision. Okay? Let's talk about vision and how vision works. So this is a picture of a flatworm. Okay, it has eyes. This is an image from like an insect, what an insect would see. Kind of interesting because they have like little compound eyes, so they would see like the mosaic of things. These are just different types of eyes. Okay, these are just different types of eyes. Kind of scary, right?
Jellyfish.
Yeah, they have eyes too. But they can't form a vision though. Like I think they can just see like where light is coming from. I don't think they can form an actual vision. But I know, like I've actually seen jellyfish chase things. So, but I don't think it's all jellyfish, only certain types. They're just kind of freaky, right? Because, they don't have like these eyes that you would think are eyes, like little black spots. Actually, I think I have actually a picture of this. Yeah, here it is. Yeah, so these little black spots are basically like eyes. And I think this is a box jellyfish. I've actually seen them chase things. It's kind of really freaky. How does vision work? So actually, one of the things that we'll do in this class is we're going to take the eye apart. We'll dissect the eye. Okay, I think we'll do it next week, which is kind of just for fun. Okay, and so this is an image of a cross section of the eye. So I'm not going to talk about how the eye works itself, because we'll talk about that next time. I want to talk about actually how the neurons work. So here's the setup for vision. Okay. These are your photoreceptors, okay? These are cone cells. These are the ones that allow you to detect color, like red, green, and blue. These are sensitive. These are tuned to color. So like, for example, this one is colored blue, so I would imagine this one is sensitive to blue light, okay? So if it receives blue light, something will happen. Whereas the red cone, if it receives red light, something will happen. Okay? These are rod cells. They just respond to any color of light. So in other words, light versus dark. Okay? Now if I was to ask you a question, if I said to you, what do you think the trigger is for a rod cell which detects light? What do you think it would be? What do you think it would be? What's the trigger for a rod cell which allows you to see light? It's what? It's light, right? Well, that's what I thought too. It's actually the opposite of that. Yeah. But before I actually proceed and explain this, is there another way to trigger vision without actually seeing light? Can you actually see light without seeing light? Can you see flashes of light or light without light being there? So if you were to close your eyes and you start rubbing your eyes, what happens? You start seeing spots, right? Those are little flashes of light. Why? Why does that happen? And you ever see like in those cartoons where like someone gets hit in the back of the head and they start seeing stars? Okay. So pressure can also actually stimulate flashes of light. Okay. Now here, the way this works is quite interesting. And I would not, when I first was looking at this as a student, I would not have predicted what I'm going to tell you. Okay, I'll tell you right now, I would be like, what? That's kind of messed up. So what we'll talk about is basically these photoreceptors are you can see they're hooked up to other neurons, so you can see the synapses, the communication between the neurons. This visual system is actually in the presence of light. So when it's light, okay, this system switches on, which makes sense, right? So the information, imagine you're seeing like something approaching you, okay? If it's light out there, you can see it. This system switches on. But the reason why this system switches on is because this actually switches off, which is very, very bizarre. So let's take a look at it, okay? This is the situation where this rod cell, okay, in the dark. So in the dark, you'll notice the sodium channels are open. Now, when we think about sodium channels opening, we think about sodium coming in, and what do you get? Yeah. Now, this is not the voltage gate, so this would be equivalent to this scenario, which I just showed you. Very similar to the way we detect smell, right? Look, sodium channel opens, sodium comes in. Sodium is what charge? Positive. Voltage goes up, and then what do you get? You get an action potential. So look, in the dark, rod cells are constantly firing in the dark. Okay? In the light, though, they don't fire. They shut down. They turn off. So I'm going to ask you to make a prediction. Okay? So let's think about it. Okay? In the light, they turn off. In the dark, they turn on. Which is like really bizarre, right? Okay. If they're on in the dark and you cannot see anything, what type of neurotransmitter is coming out of these rod cells? Okay? It's the dark. They're firing action potentials, right? And we studied that, okay? And you don't see anything. So what kind of, and I'll give you the name of the neurotransmitter. It's called glutamate. So yeah, monosodium glutamate. So glutamate, MSG, is actually basically like a neurotransmitter. What kind of neurotransmitter is glutamate then? Now, there's only two types of neurotransmitters, right? Inhibitory and excitatory. So what do you think glutamate is? It has to be inhibitory, right? So in the dark, what's interesting is in the dark, the rod cells are firing, right? They're on. They're releasing glutamate. And the glutamate turns this system off, which makes sense because in the dark, you shouldn't see anything. In the light, what happens then is the opposite. Okay? In the light, what you get is the rod cells turn off. The glutamate does not get released. And that then switches on the system. Now the system can be turned on. So I'll be honest with you, that just sounds totally counterintuitive. Right? Like the opposite of what you would predict for the way this would work. Okay. What about hearing? What about hearing? How does hearing work? So let me show you actually an animation for hearing, okay? So let's take a look. Systems, nervous system, how we hear. I think it's this. This one, maybe this one. I'm not going to show you this whole thing, okay? There's a little part here where
certainly aging accelerated by being
I'll
exposed
show you this.
cell hearing loss. All of these things will be eventual repairs of cells are.
Right here.
When sound waves enter the
So
ear
here is an animation of how hearing works. So this one is more predictable, okay? So these things are the disturbances in the air. This is what you're seeing, Okay, this is the disturbance in the air. Okay, and you can see it's causing these what are called inner ear bones to vibrate, and that's going to basically cause a disturbance here. Okay.
The sound pressure is amplified some
So
22
it's
times
causing a
as it
disturbance
passes
in this
through the
section
middle
over
ear
here,
to the inner ear,
and there's
where
a
there
fluid.
are 20 to
So
30,000.
in there's a fluid. Okay. Now the fluid is being disturbed. As the fluid gets disturbed, these what are called hair cells are vibrating back and forth. Okay. And it's the vibration of those hair cells that basically is what you perceive as sound. It's the movement of these little hair cells.
Microscopic
Okay?
hair
So that's what
cells.
you're seeing here. And that information
The hair
then
cells
gets
react
fired
sound
to what's called the
and stimulate a
auditory nerve.
going to the brain.
Okay? So here's an image of that one little hair cell. You can see it's vibrating to sound. You can see that
Under a
vibration.
microscope, you can
So
see
this is a bit more logical and and more intuitive. Like with vision, it's the opposite of what you would think, right? But here, it's just the vibration of the air generates basically vibration in these hair cells, and as these hair cells vibrate, they trigger action potentials, which then you register as sound. So like sounds that have a very high frequency means they vibrate really quickly, right? You perceive that as like a higher pitched sound, for instance. So this is more logical, okay, than vision is. The thing about the hair cells, they don't just allow you to detect hearing. How many of you have ever had vertigo, or know someone who's had vertigo? It's when you have a problem with your inner ear, and one of the symptoms is you have you can't balance yourself properly. Like you get dizzy and you fall over, because the hair cells are not just responsible for sound. They're actually also responsible for things like the positioning of the body. So they give you information. They give the brain information about like your position. So that's actually how you know your orientation is when you move your head, the hair cells will bend to one way, and then your body says, okay, the hair cells are bending this way, so I'm turned this way. Okay. So they measure other things too, like position and also acceleration. Here's the thing. I'm not saying that it's ever happened to me, but apparently, supposedly, one of the symptoms when you drink too much is that the room starts to spin or things start to rotate. That's a sensation, apparently. So I'm going to see if you guys can figure this out, okay? So what happens here is when you drink, alcohol gets into your bloodstream, and eventually some of the alcohol gets into the fluid where the hair cells are. Alcohol and that fluid do not have the same density, so when they mix, they start to swirl, okay? Now, the hair cells, well, they work because they're in a fluid, and when the fluid moves, the hair cells move, and then your brain detects that, okay? So when
one teachers are asked to keep the students until we do the call down for the assembly. Thank you.
When the alcohol gets into into that fluid, the fluid starts to swirl. So the hair cells are moving, okay? So here's the thing. Imagine you're sitting in a chair, and this is happening to you. And the hair cells are moving, right? Now, that's telling you that, it's telling your brain that you're what? That you're moving, right? That you're moving. But, here's the thing about the brain. The brain does not like confusion. Doesn't like confusion. So, what happens when your brain gets different inputs that are not compatible, that they are different messages? It tries to make sense of stuff. Did I show you that video about Stairway to Heaven?
Yeah.
When you had the information that was visual and then auditory, and then try to make sense of what you're hearing? I can show you that later if you forgot, okay? What's going on here then is what? You're sitting on this chair. Your brain, part of your brain is telling you that you're moving, but your eyes are saying, and other parts are saying, what? Yeah, I'm sitting in a chair. So part of your brain is saying, no, you're spinning around because you feel like you're spinning around. Part of your brain's like, no, I'm sitting in a chair. So what's the conclusion? What conclusion does your brain come to? Not you're spinning around, but the room must be spinning. Something's spinning, right? If it's not you, then it's got to be the room. Okay. Anyway, so I just wanted to show you some examples of like different neurons and how they get triggered. Okay. And why don't we stop there? Okay, ladies? Okay. The end.